I'll go ahead and get started and just do some of the introductory material so we have plenty of time for our speakers today. So hello, everyone. My name is Sarah Stone. I'm the Executive Director of the eScience Institute at the University of Washington. And together with my colleagues and organizers, who are the executive directors of other data science institutes across the country, I'd really like to welcome you to this next session of the Data Science Coast to Coast seminar series. This series is hosted jointly by data science institutes at 7 universities. And we've been planning ways to foster a broad-reaching data science community that can collaborate routinely to advance our sort of shared missions around research, education, and data science for social good. Our transitions to this remote setting really helped us to build this joint seminar series and provided kind of a unique opportunity for our communities to come together. And so over the past few months, we've been hosting a series of seminars where each seminar features one faculty member and one postdoc from 2 different universities. Each speaker will give a talk about their ongoing projects and motivating issues. And then so we'll have the two talks back to back, and then we'll have time for a joint discussion with the audience. And we really designed this format with the hope to prompt follow-on conversations and spark new collaborative research across our different institutions. You'll notice that the talks are grouped by themes, as you can see here, and the previous talks in the series can be found online. Before I introduce today's speakers, I wanted to give you a preview of our next month's seminar, which will be focused on using data science to explore ocean dynamics, and we'll be featuring Miguel Jimenez Uris, a postdoctoral fellow in Earth and Planetary Sciences at Johns Hopkins, and Laura Zana, a professor of mathematics and atmosphere ocean science at NYU. Okay. So moving on to our speakers for today, our first speaker is Rosemary Gillespie. She's a professor and Schlinger Chair in systematic entomology in the Department of Environmental Science at the University of California, Berkeley. She's the president of the American Genetics Association and a member of the American Academy of Arts and Sciences. Her research focuses on understanding evolutionary patterns and processes among populations and species. And today she'll be talking about data integration across space and time to infer biodiversity dynamics. Our second speaker will be Shelley Trigg. Shelley recently started a position as a research scientist at the Gloucester Marine Genomics Institute. Prior to starting this position, she was at the University of Washington as one of our UW Data Science postdoctoral fellows, and she was housed in the School of Aquatics and Fisheries Sciences. And her work really focuses on understanding how species respond to environmental change, and she'll be exploring that topic in her seminar today. So without further ado, let me turn it over to Rosemary and just encourage you're welcome to drop questions in the Q&A throughout the talks today, and then we'll address them at the end following both of the talks. Let's see, let me stop sharing, Rosemary, so that you can go ahead.
Okay, and I can go here and here, I think, and if I go to this, then I can go to, let's see where we're at. Great, can you see the slides okay here now?
That looks great.
Perfect,
yes.
Perfect. So yeah, what I want to do today is just talk to you a little bit about my work, and in particular, what I want to focus on is how we're using this massive onslaught of data that's coming in to understand questions of biodiversity across gradients of both space and time. And so the idea is to look at biodiversity in the context of the community. And I'll be emphasizing that the importance of the new data as it's coming in to answer really critical questions. So as just a little bit of background, We know the acute relevance of biodiversity across diverse disciplines and sectors. We've heard in all these news outlets about the catastrophic loss of biodiversity, the severe threats that are being incurred, and you know, that you're getting insect decline and all of these species going extinct, and so many reports about just what's going on in the world. So what we really need here is some understanding of how change is happening. What I'm going to do today is talk a little bit about we'll focus on entire communities and threats to these ecosystems, and try and see how we might thread together different kinds of data to understand, to get an understanding of the health of the planet. And so try and find quantitative ways of understanding what is actually going on. We really don't have any way of just looking at a system and looking at an ecosystem or a community and saying, you know, this one's really doing okay, or this one's in real trouble and we need to intervene to keep it on track. So what I'm going to do is just go through three aspects of my work. I'll talk about using different kinds of means through which we can get at dynamics. We always need to look at some kind of time frame in order to understand change. So I'm going to first of all focus on island chronosequences, which are just geological time slices where you can look at communities of different age. Then I'm going to talk about museum specimens through time. And then what I'm going to do is just talk about how you might integrate these data types to look at the dynamics of change. So just to start off with, I'll go quite quickly through the island chronosequence work that we've been doing a lot of in my group. And the focus here is we're looking at the Hawaiian Islands and looking at signatures of ecosystem change through time. And the thing here is that you've got this island hotspot, and the most remote island group in the world. The Hawaiian Islands are disproportionately impacted by anthropogenic influences because they're so isolated. They've got such a uniquely endemic biota. And so because of their small size, their isolation, and their vulnerability, they can serve as kind of harbingers of what can be expected globally. So what we've been doing is looking at this sequence of islands in the context of as I said, a chronosequence, slices in time. This is possible because of the hotspot nature of the islands. They emerge from down where the current highest island is at the moment, the island of Hawaii. And so they emerge there over a hotspot. And then over time, they go away on the Pacific tectonic plate. They get carried away. And so the oldest island is Kauai. So basically, you've got this series of islands going from the youngest island on the big island, the Maui, Molokai, Oahu, and over to Kauai. And so you can use this sequence to look and see what are the characteristics of communities that are just starting on these new lavas, and how does it change through time? And in particular, how do these attributes of the communities affect resilience and whether they can accommodate disturbance and response to invasive species? So just a very quick summary of what we're doing. We're getting empirical data through what's called metabarcoding, where you just generate lots of genetic sequences from every individual in the community. And it serves as a kind of ecological tool for understanding diversity in the system. And so you can use that data. You've got data for everything in that community. And so you can use that data to understand the properties of the entire community. So their abundance distributions, who's interacting with whom. And so you can understand the properties of each community as you go through time. And so you can use these systems to look at functionality of the system, robustness or resilience to change. And just to give you an idea of what we've been doing, so you've got this chronosequence going from the youngest island up to the older, and to the oldest island on Kauai. We've got these little spots that we're sampling. And so we sample there. And just to give you a feel for what the data is, so we sample in different parts of a given site. We're focusing on arthropods. So you just sample in all sorts of ways at a given site. And what do you get? You get lots of samples, you get lots of data, you get lots of specimens. And these are arthropod specimens that we're focusing on. And you could just get thousands, millions of them. And what do you do with all that data? Because you've got so many sites, you can't possibly go and identify everything and catalog it. So what we do is we simply sequence it, and we get genetic sequences. And this is a work that has been pioneered by Henrik Krehenwinkel, who was a postdoc in my lab. And so what Henrik has been doing is just generating this data from the different plants, so getting associations with plants, dividing it by size, and then you get these amplicons or amplicon sequence variants that can provide information on what genetic sequence is associated with what plant and how they interact with that plant. And so it gives you all of this data, and a lot of data. And the point here is that with this massive amount of data, what we can start to do now is look at these signatures of change through time. And so what we've been doing is looking at identity and abundance of arthropods through time. We're looking at networks of interactions, who interacts with whom, and hence that allows us to look at, is the community more vulnerable to invasive species? We can also use the data, of course, to look at how many species are native and how many are not native. And so we can look at these interacting effects. And just to give you an example here, you can look at how the network structure changes through time across this Cronus sequence. The expectation is that the youngest sites are going to have very simple networks, the older sites very complex. And is this complexity associated with more resilience to invasive species? These are the kinds of questions that we can get at with this massive amount of new data. So what other kinds of questions can we get at with this kind of biodiversity data? And so what I want to do here, I've talked about this, what I want to do is just talk about the other kinds of data that you can get at looking at change through time. And so I want to bring up here the importance of museum specimens. Now, museum specimens have been used in the past a lot for understanding change through time, but it's only recently that we've got massive opportunities for understanding these museum specimens in very different ways. So the museum collections provide a physical record of past environments and past adaptation to those past environments. So just for example here, this is a work that a colleague, Craig Moritz, did up in Yosemite, and they were looking at these chipmunks, and the alpine chipmunk here at the top it's a high elevation specialist. And what they found is that 100 years ago, the initial director of the Museum of Vertebrate Zoology in Berkeley, he collected some of these specimens. And so that the specimen was in the collection. And they went back and resurveyed that site, found more specimens. And so they looked at this chipmunk and said, well, has it changed? And so they found first that the range had contracted, presumably due to climate change. But they could also look at the genomics. And so they could look at the genomic signatures. For example, this is a chipmunk speciosus. It hasn't changed its range. And what they could see is signatures in the genomic makeup of the retracting range of this particular species, Alpinus. And what's also really interesting here, and what they were also able to show, was this, they were looking at the genetic covariance between the historic specimens and the current specimens. And what they found was there's strong covariance, but you also get outliers. And these outliers are showing the regions of the genome that are being subjected to selection. So basically what this is showing is all of this new data on genomics is allowing us to understand how organisms are changing through time. And we've got other avenues to use these specimens through time. Looking at bees, for example. This is work of a colleague, Todd Dawson. What Todd was interested in was looking at the pollen that bees carried around with them. And so using specimens in the collection from, the beginning of the last century. And so looking at the pollen and just using it as a kind of sample of the environment. And so what they found, this is samples of the bee pollen in Berkeley. And you go from 1937, 47, 57 to 87. And you see the isotopic signature has changed, indicating that the environment that the bees are sampling during that period has also changed. And there are other ways, of course, that we can use these collections to look at change, to look at the dynamics of change. For example, this is work that was done by David Ackerley and colleagues here at Berkeley, looking at how the environment has changed in paleohistory, and looking at pollen records that you can gather, recovering the history of the environment, how the environment has changed, and hence how fire has moved in and modified the environment since human arrival. And so we can record all of these events, we can understand these events through this dynamic reconstruction using this specimen data. Now, I've gone through this quite quickly because I want to emphasize the critical part here is that you've got now very diverse data to assess the dynamics of global change. And so we've got different kinds of data, for example, the specimen data, they're point data, but we've also got layers of data. And we need to connect these in order to get a holistic picture of how things are changing. So what we've been doing is trying to see how we can integrate across the collections and historical data that we have, integrate these with environmental layers, and use models to better understand land use, how it's going to change, understand the synergy of land use and climate change and biotic response, and also be able to translate the findings for NGOs and identify areas that are affected by rapid change, and then go on to develop policies and strategies. So we have to be able to connect these different kinds of data in a manageable, in a sensible kind of way. So a few years ago, some years ago now, with funding from Keck and the Moore Foundation, we were able to develop this approach for integrating disparate kinds of data. This was a very preliminary attempt at trying to connect all of these data sources. But the important thing was that we recognized the very marked differences between these data types. And the important thing was connecting them and integrating them to make a more manageable kind of structure within which you could ask questions of global change. So what we were doing then is getting this data and integrating it with all of these environmental layers and going back through time so that we can understand the dynamics of change. Because unless we understand the dynamics of change through time, we're never going to be able to develop predictions into the future. So where are we now? What we're trying to do, as you know, you got these increasingly massive new data sets coming in from all sorts of sources. We're trying to use these to see if there are ways to integrate these very diverse and broad and huge data sets, so that GBIF data, BOLD data, these are all genetic or specimen databases that we're trying to link those to ecological data, link those to online environmental data layers. But then the big thing is interpreting and visualizing the data in a way that can be understandable. So this is where I'm working very closely with BIDS, in particular, Cody Markles and Sierra Martinez. And what we're trying to do is understand what the dynamics of change in biodiversity and how this might be translated into information that's relevant to health, fire mitigation, agriculture, etc. So the thing here, just to kind of think about what kinds of questions you can address here. We want to be able to deliver the data in a standardized form and serve the functions that just allow stakeholders to actually access it and be able to interpret it for themselves. And so what are the kinds of questions? I mean, just to show you here, I'll just give you here in the top one here, just a couple of examples of disease outbreaks where they use biodiversity information from museums to understand how disease has changed, like hantavirus. The museum specimens were important for understanding how the populations had changed, and so basically to predict the hantavirus outbreaks. And likewise, guinea worms. Museum collections have had a long history of importance in disease outbreaks. But coming to the present, we've of course got influenza being really critical. It was really critical to have previous collections to understand the 1918 Spanish influenza outbreak. The collections allowed us to reconstruct the virus history and how it had changed. But the thing now, we've got COVID, and why are collections, why is it important to have the data that I'm talking about to understand COVID? It's because we now have a lot of data on the vectors, on bats, on other disease vectors out there, and also how the environment is changing. So we can use the combined massive data sets to understand how basically the change in the human-wildlife interface, which is really the #1 cause of the initial cause of the COVID pandemic. So importantly here then is that we need to integrate this data and in order to understand what's going on. So as I said, just to kind of wrap this up, environmental change problems are increasingly complex. And so it becomes incredibly important that we integrate across scientific disciplines and their data. The biggest thing here is that we've got to connect across these very disparate areas of biology, but not only biology, into social sciences and into politics, and really work together in the context of data science. And just as an example here on one that I'll finish off with, fire. This is a huge issue, not only in California, but in Colorado, in other parts of Australia, massive problems as well. But it's obviously a huge issue in our world at the moment. And there are many things that are contributing to these fire outbreaks. Importantly, a lot of them are biological and things, relying on specimens that are held in museums. And there are also specimens that we can go out and collect and examine, but we need the data. We need the data from the past to see what was there. We need the data from the present to understand how the current biodiversity is going to be responding to the fire and to the changed environments. And so we need to integrate these very disparate kinds of data. And Cody Markles, who recently joined the BIDS community as an environmental change fellow, is doing exactly this. So Cody is in his just 1 month of being at BIDS. What he's looking at is the data associated with the fires. So looking at using the UC reserves, many of which have burned in the recent fires, and looking at the environments that were there, the environments that are currently there, in sites that are totally burned, less burned, and looking to see how the biodiversity has actually changed using metabarcoding data that I was talking about in the Hawaiian Islands, using sensor networks that are looking to see what's going on in the environment currently, and then putting all of this information together to predict future species distributions, future fire risks, and what interventions that we can actually achieve to make a difference, and how we can prevent fires moving forward. So this is just an example here, but just to summarize what I've been talking about, what I've tried to show you here is how we can use very different sources of data to understand biodiversity dynamics. We've got to understand biodiversity dynamics in one way or another, whether in deep time through these island chronosequences, or through recent time, whether it's looking at museums, specimens across 100 years, or looking at change in as a result of fire in the last year. We've got to have a trajectory, a timeline, and we can get these massive amounts of data from eDNA, metabarcoding, all kinds of genetic genomic data, integrating those kinds of data with environmental layers and with serving them up in a way that's accessible to stakeholders. This is going to be our major challenge as we move ahead. And so with that, I just want to thank everyone, and I'd love to have more discussion later, but for now, I'll just hand over to Shelley, who's going to be talking to you more about marine genomics. And I will now, if I can figure out where my cursor is, I will, stop sharing so that Shelley can go ahead.
Everyone see? Good.
Yes, looks good.
Great. Okay. Yeah, so that was a perfect segue into this. Because I'm gonna be focusing on these genomics data sets and also talking about how we can integrate them. As Sarah said, I'm a former University of Washington data science postdoc fellow, and was in Stephen Roberts' lab at the School of Aquatic and Fishery Sciences. And so I'm going to be talking about some research I did there, and also some research I did during my graduate studies, and with the NOAA Northwest Fishery Science Center. And I just wanted to give a little plug to where I'm at now, the Gloucester Marine Genomics Institute, which is fairly new. It started in 2013. It's a nonprofit research institute, and the research department started in 2017, and we're very interested in researching ocean, environment, and human health challenges. And all right, and I wanted to acknowledge the funding for the projects that I'm going to talk about, which are shown at the bottom of the screen here. All right, so I'm going to give some background on environmental change and omics, and then go through some exposure experiments and omics analyses I performed on different animals, and then summarize the different responses that I've seen and talk about these future directions. Okay, so we know that global ocean change is happening, and that over time we've got this increase in atmospheric carbon dioxide that is correlated with increase of ocean carbon dioxide and a decrease in ocean pH, and leading to this ocean acidification problem. And we also see ocean deoxygenation, which is co-varying with pH, so lower oxygen with lower ocean pH. And then we've got ocean warming happening with the increase of carbon dioxide in the atmosphere, and then ocean salinity variation resulting, so with melting ice caps and glaciers and such. So there's this big question how climate and ocean change will affect different animals, especially as things progress into the future. And we don't really know what's going to happen with biodiversity. And I guess in terms of studying how different animals are tolerating these different conditions, we can study them in their natural habitat, or we can take them back into the lab and put them in these different conditions in controlled experiments, and look at different processes in each animal. But it would be really nice to be able to look at similar processes, especially if they're shared ones across a number of animals, to be able to compare species. And so one way to do this sort of in a systematic systems biology way is to look at all these different molecules in the cell. So the cells, all animals have cells, and cells are like cars, and that they have all these individual parts that make the system run. And yeah, so like I said, like all animals have these basic cellular parts. And if we know how these are working together, we can more easily compare across different animals. And we can get that kind of systems perspective by performing different genomics analyses. So omics is the study of collections of biological components. So like the genome is a study of all the, is the genes or genetic material in the cell. And then there's the transcriptome, which is RNAs, and then proteins are form the proteome. The interactome is how all the proteins bump into each other and drive biological processes. And then the outcome of these processes, there are these small molecule byproducts, metabolites, and the collection of those is called the metabolome. And these components are analogous to how a meal is prepared, and that the genome is really like the recipe in the cell. The transcriptome or the RNAs are really like the orders being called across the kitchen. And then the proteins are similar to the sous chefs who all bring their special individual skills. And then how the chefs interact is like the interactome, and how they use their special skills to work together to make a soup or grill a steak. And then at the end of the day, you get this metabolome or a meal resulting from everything. So now I'm going to talk about these different experiments in the omics analysis. And I'm going to start with the metabolome response to ocean acidification conditions. And in this experiment, we looked at Dungeness crab larvae that were metamorphosing into juveniles. And they were reared in these different ocean conditions, so low oxygen, low pH, and then a combined treatment for about a month. And there was no effect on their survival or their development in these different conditions. So we wanted to look at what was going on in their metabolomes to try to understand how these animals were physiologically dealing with these conditions, what enabled them to survive fine. And what I did here was apply different statistical approaches to identify what metabolites were changing in the animals that were in these different treatment groups. And so what we're looking at here, each shape is a different amino acid, because the amino acids in general were more affected than lipids or sugars in these animals. And if the shape is blue, that means it was lower abundance. And if it's yellow, it means it was increased abundance. And so what you can see is that in the low oxygen group, there was more amino acids that had a higher magnitude of change than, for instance, in the low pH condition. And when you look in the combined stress condition, you can see that it's really similar, like the trends are very similar to the low oxygen, indicating that the low oxygen has a more dominant effect on these molecules. And what it means in terms of biology, so these shapes are all arranged, they're clustered by their chemical similarity, and they're connected by these lines, which you may not be able to see, but those lines indicate they're in a shared pathway. And these molecules that were changed in the low oxygen condition were related to energy conservation pathways, where in the low pH condition, they were more energy generation pathways. And then in the combined stress treatment, they were doing more conservation. So we kind of took this all to mean that the crabs are really maybe when they're in the low oxygen, they are able to kind of hunker down and use less energy and still survive. And then under the low pH condition, maybe they're needing to generate more energy to fuel the machinery they need to maintain cell pH levels. And so now I'm going to talk about a very similar experiment. This is on pteropods, which are these really small mollusks that float around in the ocean, and they eat phytoplankton and support ecosystem health. They're a prey source. Fish eat them. So we raised these animals in the same conditions, only for a shorter period of time, because they just don't live as long in captivity as the crabs do. So we did a shorter experiment here. And again, there was no effect on survival. So these animals are able to tolerate these conditions for this duration. And again, I performed the same multi-statistical approach. And this time identified that in these animals, it's lipid metabolism that seems to be affected. And the low oxygen conditions tended to increase certain lipids. And in the combined stress condition, it largely increased these phosphatidylcholine molecules, where in the pH treatment, it tended to be more sugars. So again, we think that the sugars are more related to generating energy to fuel pH regulation, and potentially this increase of these particular lipids could be a way that their cells are changing their membrane permeability so that they can allow more oxygen inside the cells. But again, we see that there's this dominant oxygen effect more than the pH. And now I'm just going to go through the proteome response to warming. So these are the sous chefs, so doing the work in the cell. And this is looking at oyster and how they respond to warmer temperatures. And these are oysters that are also transitioning from a larval to juvenile stage. And the two temperatures we looked at were actually warmer than what you would find in the wild. These are more relative to culture conditions. This is work that was done at Taylor Shellfish Hatchery in Washington by Rhonda Elliott. And so what they were seeing at the hatchery was at the lower temperature, at this 23 degrees, that the animals were experiencing mass mortality. So actually increasing the temperature was allowing the animals to survive better in the culture conditions. So we wanted to see why that could be. And then in this experiment, there was no mass mortality, which was good, made it easier. We were able to just compare their proteomes without it being confounded. And then the oysters that were in the warmer temperature did tend to be larger. And so we did proteomics to measure the physiology, and I performed a multivariate statistical analysis to identify what proteins are leading to these, could be leading to these differences. So here you can see the different groups. This was a time series data set. So there's different groups and different time points. And you can see that this analysis was able to tease apart the temperature effects here. And here we're looking at a heat map where each row is a different protein. And if it's the color is red, it means it was a higher abundance. Blue means lower abundance. And you can see there's two different trends going on here, two different major trends. And one is that there's proteins that are in lower abundance in the higher temperature group, or there's proteins that are in higher abundance in the higher temperature group. And what those proteins, the processes that those proteins were involved in, so the ones that were in higher abundance had to do with different growth pathways, which makes sense because these animals were larger in size. And the proteins that were in lower abundance had to do with immune response. So an immune response that may have been activated at this lower temperature, but not needed at this higher temperature. And that led us to think that there could potentially be some microbes growing in the water at the lower temperature that were not present at the higher temperature. And so there's some follow-up work looking at that. And I'm just going to go through this one last experiment, looking at the methylome response to environmental change. So the methylome, I'm talking about DNA methylation in the genome. So these are marks on the DNA that alter the phenotype or the performance of the animal, and they can be heritable. And they also can be induced by the environment. So changes to DNA methylation can be induced by the environment. And I'm going to be talking about this in Pacific geoduck, which are these big burrowing clams native to the Pacific Northwest. And they're a valuable fishery there. And they're a biofilter and prey source, and also important to Native American tribes there. And this experiment is looking at how they respond to seawater pH. And so in this experiment, the animals were exposed for a couple weeks to low pH, and then all put at the same common ambient condition for a few months. And then they were exposed again to low pH to see if this initial exposure might impact how they respond, and especially at how their methylome responds. And this is an experiment that was done by Holly Putnam, who's a professor at University of Rhode Island now. And this is some work she did as a postdoc in Stephen's lab. And she saw that after the initial exposure, the animals that were exposed to the low pH treatment tended to be smaller in size, but then after the months without seeing the exposure, they actually had this compensatory growth. So if they initially saw the pH, they grew larger than animals that never saw it. And then upon the second exposure, animals that had never seen the pH, the low pH before, exhibited delayed growth. However, if the animals did see the pH before, they didn't have that. So it sort of protected them from that growth delay. And when we looked at the methylation, we found that this is kind of like in general across all the animals, the methylation tended to be in genes. And we each performed different approaches, multi-statistical approaches to identifying differentially methylated regions and genes. And here we're looking at on the left side of these heat maps. So each row is a different region or gene on the right side. On the leftmost column in these heat maps is the ambient condition, and the other two columns are the pH treatments. And if it's red, it means there was more methylation, and blue means less methylation. And so where these regions were in the genome tended to be in or around genes, and happened to be genes that were involved in cytoskeletal activity or nucleic acid binding and mitochondrial activity. And from the differential gene analysis, there were specific biological processes that tended to be affected. So one, interestingly, after the initial treatment, so there's an acute pH effect, and also this longer lasting effect on Wnt signaling, and it did happen in different genes in this pathway, and Wnt signaling is related to growth processes. So their animals are able to alter the methylation in these pathways that is associated with this difference in growth that we see. So then I'm just going to quickly summarize all this diversity in these animals' responses. So as you can see, there's we've got different species. There's also a couple other experiments that I threw in here that I didn't talk about. We've got all these different species. There's different conditions. There's different lengths of time, and there's different life stages. And then most of these experiments, or I guess, yeah, all of these, there was not survival differences, which kind of helps that. These are more like kind of, I guess, a base, give us a baseline. And then there's all these different omics data types, and then different molecular phenotypes. So like thinking towards the future, it will be, now that we have all these data sets. So those are just data sets that I worked with. There's hundreds of other data sets available for different species and different with different phenotypes, and it's now a great time to be able to integrate these, to be able to glean more from that, especially in terms of how these animals are going to survive in the wild. So not just in the laboratory setting. And it would be so great if we can use these to predict how these animals respond. So you could take animals that were, under these different environmental conditions, either from like field measurements or in laboratory setting, and be able to integrate all these different data types, and then identify different marks that are either leading to environmental tolerance or intolerance. And these marks could be, like particular genes that are common, in different animals, or marks on the genes, or it could be different biological processes. It could be like one step higher than that. So maybe it could be different genes in different animals, but a common biological process that is happening to lead to tolerance or intolerance. And this would be super, super helpful for being able to do biomonitoring, so being able to do early detection of disease or pollution in different animals, or just predicting how they're going to migrate. And yeah, so just being able to compare across different species. And then I guess kind of one example of how we might do this is by taking different data sets and systematically reprocessing them and using standardized pipelines. So integrating genomics data, methylation data, RNA-seq data, proteomics data, and even metabolomics, and then getting a common output that can be compared. So for instance, like counts. And then you take your count data, you could, one approach is to analyze each individually, sort of as I've shown, each of those analyses were done separately, comparing the controls in the experiment to the animals under treatment, and then do a post-analysis data integration. And another possible option is to take all the count data and perform some correlation modeling or network modeling, and doing this integrated data analysis. And then the outcome would be something like, this is just one idea, like a biomarker database. So where there could be markers that are associated with like maybe early signs of poor performance, or animals that are being energy efficient, or animals that are not being energy efficient. So thinking about marine omics for future biomonitoring, there's a lot of promise here in that the genomics technologies are quick, cheaper, and more efficient at the data collection. And we get a lot of data very fast compared to traditional methods. And the omics data allows a high resolution, very high resolution answers to different research questions that we have, and may lead to more sensitive and accurate biomonitoring. But as you can see, there's different challenges, like these many different factors that would go into different models. And then there's different sequencing technologies that are used, but those could potentially be normalized with commonly measured molecules across the different data sets. And then there's different data quality, like depending on when it was done, and as things progress, the technology gets better, but thresholds could be applied to based on the internal standards that were included in those experiments, to make the data sets more comparable to each other. And then, you can imagine there's a lot of data coming from here. So there's a data storage and management challenge and computing power needs. And then lastly, this kind of work requires integration across different fields and disciplines, but that I think is also something that's great because it's makes it very ripe for collaboration. And yeah, so on that note, I just want to thank the Roberts Lab, Stephen Roberts, and all the Roberts Lab members for all their help with this work, the Putnam Lab, University of Rhode Island, University of Washington Genome Sciences, Emmeton and Schiffman, who helped with the proteomics stuff, and then the NOAA Northwest Fisheries Science Center folks. And with that, I'd love to open this to discussion.
Thank you so much, Shelley and Rosemary, for those wonderful talks. And please, folks, put questions in the Q&A. We've had some actually come in, and Rosemary, please feel free to unmute yourself too, and maybe you guys could both respond. There was a question that came in, so started off saying, thank you for the presentations. It seems like you both are speaking to the difficulty of not only having data to measure environmental change over long periods, but also in having agreed upon definitions of environmental health. Do you think that there can be one overall metric, or are you even aiming for that? Or are you thinking there will be different metrics in different contexts? And I'll just open it up to both of you to answer that. I know Rosemary provided a bit of an answer in the text there, but I think it'd be nice to share out with the full audience.
Yeah, that's an excellent question, and it's something that we've been playing around with a lot. And I'm actually working with others at City University in New York, and in particular, this guy, Mike Hickerson, and Isaac Overcast, also Andy Rominger, who's at the University of Maine. We're trying to figure out whether there are signatures in communities as a whole. Is there a signature of genetic health? So where you sum up the genetic distances, either between close relatives or genetic diversity across the entire community, is there a signature there? Don't know the answer. You can get signatures from abundance distributions, because abundance distributions tend to follow very discrete laws, and so you can see when a community is out of whack or when it's not. And so you can use that to say roughly what's going on, but you really can't, there's no mechanism there. One thing that we've been focusing on fairly heavily is network structure, and trying to see whether the metrics you can derive from network analysis tell you about how robust the community is. So for example, when a community has just been completely, when it's just been wiped clean, you tend to get one thing coming in, and so that there are very few interactions. But as the species build up, you get more balanced interactions. But then if an invasive species comes in, it dominates the entire thing. So can you pick up those kinds of signatures? But yeah, that's an excellent question. Over at Shelley, what do you want to add there?
Yeah, I'm thinking that it's different metrics in different contexts, and I think, yeah, I think in these laboratory experiments and for like particular, durations of like environmental exposures, it can be different when you're looking at the animals like in the wild and looking over a longer period of time. But I definitely still think it's there may be the signatures of like when the animal health is going poorly. So like the data that I showed was the survival for animals where the survival wasn't affected. But I mean, if you're looking at animals, you know, that have different or have more negative response, there could be some signatures there that could emerge from doing these meta-analysis.
So, you know, I just to point out one thing here that people might have noticed, you know, because what Shelley is talking about, which I think is super interesting, you know, response of given species to change, which is so critical to understanding, well, how organisms are going to respond in the future. What I've been talking about is how whole communities are responding to change. And so the critical thing in terms of data science and a huge challenge is putting those two together, because we have to be able to understand both the community response as well as the species, population, individual response. Anyway, I just thought I'd throw that in there.
Yeah, I was really thinking about that too, and in the context of both your talks, and I was wondering about what you both experience as the sort of the biggest bottlenecks to that type of data integration, to bringing those disparate data sets together. I guess in terms of like from a data science perspective or from other types of perspectives, what are the biggest challenges in this space, do you think?
I think it's basically not, it's really hard to do, because it's a very, you know, the community data is shallow and broad. And so it's got this big spatial component. You're looking at all these signatures of the abundance and the, you know, who's interacting with whom and all of this kind of stuff across the space. And then you've got this, the information on the lineage, which is basically channeling down into, you know, the response of that lineage within the community. And it's what the challenges are, well, I mean, it's just a different, it's data that's kind of orthogonal, and it's a challenge. And this is something I've talked to Cody Markles, who I mentioned in the talk, as to, you know, how could you possibly do this? And he's been thinking.
Yeah, it seems like you'd have to like reconstruct communities or something like that, and take, you know, be able to take certain ones out in a controlled way to try to understand the interactions potentially. I don't, yeah, because it's tricky.
And I know we're coming up against time, but there was another question that came in that I thought would be a nice way to answer. And that's about, you know, given the complexity of today's data and methods for analyses, as well as the importance of biodiversity dynamics for so many issues relevant to society and things that we're certainly facing, for example, in the context of climate change. How can we best communicate about this topic with the general public? And I know this is something that probably both of you have thought about or have experience with. So if you want to, maybe we'll wrap on that, because I think it's always nice to come back to how do we take what we're doing in, you know, more of the academic realm back out and integrate it with the information and communication to the general public.
Well, so I've got an advantage here in that I saw that question before. So I'll give Shelley a chance to think about this, because it's a super challenging question. And so the thing there, I think, is that you have to include the community, the policymakers, whether resource managers, the general public. You have to include the stakeholders in the conversation as you develop these structures, as you integrate across data streams. You've got to include the stakeholder in that conversation, because you've got to build something that the stakeholders can use and that they want. And I think too often we tend to build something thinking, oh, this is what everyone wants, but then no one uses it, and which can be super frustrating. So I think the critical thing is to include the stakeholder every step of the way. Over to you, Shelley.
Yeah. That is a good question. I definitely agree with that, like including the community and the management and everything. And especially, I mean, even people that are in like commercially invested too, I feel like it's so important to get everybody on the same page about, you know, like what we're seeing and what, I don't know, the potential of these tools and these analyses. Yeah, I mean, I definitely think, you know, trying to stay open and not like fear provoking is very important, or not to be too complicated and get bogged down in detail, like, you know, like too technical. But yeah, I think it's really important to kind of stay at a level where everyone can kind of understand all these different aspects of this kind of data science. And
yeah, and I'm sure that came into your work, Shelley, with because you know, you touched on a number of kind of commercially important species, and so there's definitely, you know, very complex dynamics in thinking about maintenance and management with those species, particularly as we move into a changing climate. So, well, I'd love to thank you all for joining us today, and thank you, Rosemary and Shelley in particular, for your talks. And I think that they intermingled very nicely, and as we talked about, touched in different areas, but really complemented each other well. And I think I left with having an idea of how we might bring those together and put them together in a way that could really help us think about how we make predictions about what things look like in the future, and how we start to approach that with very complex and disparate types of data. So thank you both, and I'll invite you all to join us for our next session on June 16th, where we'll be focused on ocean dynamics, which we got to touch on a little today, or at least some ocean creatures, but we'll build on that next time. So thank you all for joining, and see you then.
Thanks so much.